Hi guys. I thought I should give you a bit of a podcast on what I want you to do with microscopes as a reminder. So here I have one of my microscopes from the Year 12 lab, and of course I explained to you had to make sure you had a hand under it when you're carrying it and a hand on its neck. This way, that way. So you clearly got good control over the microscope before you move it anywhere. Of course I've taken the dust cover off, and when I'm finished I'll put the dust cover back on, but we want to see the bits, don't we? So there are some wonderful features to these particular ones, and I'll try and make that a little bit better for you to look at. Another way, that way. So as you can see, we have an eyepiece. We call this the ocular. Oops, the ocular, and it's 10 times magnification. Am I getting that in the camera? No, there it is. 10 times magnification. And then I have 3 objective lenses, and these are the ones on this dial here. The bottom one with a red ring is a 4 times ocular. In fact, you can't see it because it's now on the other side. So that's that one there. This one here is 4 times, it's got a red ring. I always start from that 4 times one. The next one round is 10 times, it's got a yellow ring. And then the 40 times has a sort of a green ring. And if your slide's good enough, you can get closer. You'll see there's a spare spot there too for another one. And often we have 100 times oil immersion lenses. And so you have a slightly different technique, which we won't have to worry about just yet. So what else can I do with this microscope? Well, you can see my hand here, it's gone to the coarse knob. I can raise and lower the stage, that's this part here, with the coarse knob. And then having got close enough, I think it's pretty good. On the outside of the coarse knob is a smaller knob for fine focus. Now we can't actually see the stage moving, but it just moves a little bit to let me get a bit closer. Really handy. This has its own light source, which is not working. That's handy. Just plug this in. Wasn't plugged in properly. So there's a light source. You can see the light coming through down the bottom there. Yep. So the light comes up through the sample I'm looking at, and I look down to the magnifying glasses, these lenses, and get a much closer view of what I want to see. With the stage, I can actually move what I'm looking at around. So with the little knobs underneath the stage, I can move the stage. Actually, I'll turn that light off, it's a bit there, isn't it? I can move the stage across. You see the silver thing going along. So I can move my slide that's trapped in there, or I can move it up and down, and the whole stage moves up and down in this case. So that means I don't have to touch the slide when I'm trying to focus on something I want to look at. So that's my microscope. Now, I've asked you, of course, to make me some things called wet mounts. So I'll quickly demonstrate what I mean by wet mount again. And I've got, oops, I should probably put that down there, don't I? So I've got my little beaker of water with a pipette. Oops, too close. Sort that out somehow. My glass slide, microscope slide. I've got some cover slips in a little container. We only want one, so I'll take that out in readiness. Where are we? One of it. There it is. Put it on top of there for a minute. And I've also got some iodine. I've got some iodine with me, so I can show you how to stain it as well. So I've got some iodine stain. This will stain for things containing starch. Turns a nice, lovely blue-black colour. And as I was coming back into my office, I grabbed a leaf. Now, we're not going to get a very good microscope sample from this, but so you can see it on the camera, I just grabbed a nice big leaf. As you can see, far too big for my slide. So, to start with, I want to mount my sample. So I'm going to take a small piece. I should have a scalpel to do this a little bit better, but I didn't grab a scalpel, I should have. So I want a small piece of leaf. This is already too thick to see much under the microscope, but for what we're doing is fine, because you want to see what's happening. So I've placed that on my microscope slide. It's a wet mount because I'm going to put some water on it. So it's now got water over the sample. That keeps it hydrated while we're looking at it, which helps. And I then take my cover slip and I drop that. It's a bit hard to catch, isn't it? Drop that down. It's now trapped. I'll try and tilt this without making too much of a mess, especially water on my computer. That's now trapped between the cover slip and the slide. So I have what we call a wet mount. And that wet mount can be placed under my microscope, which I'll do when I move everything out of the way again. So I bring the microscope back over, and I can put that down on my stage. I move this little lever here out the way. In fact, you're not really seeing that. I'll tell you that in a minute. Slide my slide into the space there and close that lever. So hopefully now you can see that I've got my slide trapped between this piece of metal here and the piece of metal on the other side. And now when I move my stage by those little knobs I showed you before, the slide moves across the stage. I can do it very slowly and watch things. So then I want to see what's going on. I want to have a look at something. So I start with my light on, of course. So I want light coming through my sample. In fact, I'll show you what that looks like now. It looks much better now. Hopefully, oh no, it's okay. There we are. Hope you can see the light coming through the green leaf. I take my slide, my microscope. I can look down my ocular, and it's not in focus. In fact, my sample's not even in the view. So I'm going to move my sample across, so it's now in view. Where are we? Oh, we're going the wrong way, I think. And there it is. So now I've got something in the way. There's a big green thing in the way, but it's not in focus. So I focus that in on 40 times, and I wish I could show you what I'm looking at here. I wonder if I can. Oh, I'll be beyond my technical abilities. But anyway, I've got this lovely green leaf here. I just finally focus it. I can actually see striations across the leaf and veins and all sorts of interesting things on the leaf surface. And a lovely donut beside it, which is my air bubble I trapped. So I didn't do a very good job of this one. So I've got a really lovely view. Now this slide isn't very good for use for much at all really, but I can take that round to 100 times, because now I've got it in the middle of my view. I can move these around and I always look at the same thing. The center of the view stays there. Oh, don't even need to adjust it. Beautiful. And now I can see some interesting little colorations. There's little something infecting this poor leaf. Anyway, I've got a lovely little view now of the cells. This won't give me a good view at 400 times because it's not thin enough. So I'll bring my next objective into place. Oh, yeah, no, it's too thick. Too thick. Not going to get it to focus as well as I'd like. But if I happen to, oh no, it's coming up. Yeah, it's a little bit fuzzy. It's not too bad. But if I'd managed to get it to be one cell thick, then I'd have a really clear view. Now, only there's one thing to do to it. This cell I wouldn't bother staining normally, but let's say I was searching for something and my stain would help me to get a better view. I can take this slide now and I can stain it. Now I'm going to stain it with iodine, just because that was at hand. My year 12 had used it last period. And I've got some iodine. Let's see, a little dropper container here. And I've now got iodine in this little part of the dropper. And I don't need very much. And what I'll do is I'll drop a small bit above the cover slip. Oops, not too high enough anyway. Above the cover slip. I haven't removed the cover slip, still there. And the stain will slowly move down through the leaf. And you can see how it's staining quite quickly. We've got a visitor in the background. And that's now stained. So I've made a wet mount and I've stained that wet mount. And hopefully that reminds you about what you need to do when you're using the microscope. And I'll talk to you again another time.